Okay, proteins. So real quick difference. General molecular formula of carbohydrates. What is it?
C6,
So
H12.
C, I'm gonna
C1.
adjust it
Ratio?
to,
Ratio, oxygen.
O1.
yeah. Because it's a variant of this, because sometimes it's C5, for example. What about fats, lipids, for what we talked about in this class? Molecular formula.
Oh.
There's
C,
technically more, but.
H, C.
So C, H, O. A different comparison, there's a lot less oxygens,
A lot more hydrogen.
a lot more hydrogens.
Yeah. Carbon.
So different variant, but CHO. How about
It's glycerol.
proteins?
Bro, I know this one.
Carbohydrates, lipids?
Oh, it has an acid group. That might the structure.
It's H, hydrogen,
So we've
nitrogen,
got-
carbon,
It's the same.
We got carbon, we got hydrogen, we got oxygen. Do we got anything else?
and then we got
So
yeah,
big
no,
one.
nitrogen.
Nitrogen. Very important. Proteins are made of amino acids. If you've taken nutrition, that should ring a bell. Amino acids are made from amine groups. Amine functional groups are nitrogens with 2 hydrogens coming off of them. And since they're amino acids, they also have
And it's the acid group.
an acid group, which is it carboxylic acid? Is it?
A double bond, oxygen and OH.
So the ones we wrote on top, those are the ratios, or is that the actual?
Those are the.
This is a ratio. These are just elements that are within the molecules.
Oh, so it's not, that's not the,
Yeah.
not the,
ratio.
Amino acids. So the proteins also you'll see is things like sulfur and phosphorus and a few other things. There's a few other exceptions in different places, but these are the general things that are talked about. Okay.
and that's the N, nitrogen with 2 hydrogens, that is the mean amino acid with
The amine of the amino acid and the acid of the amino acid. This is able to donate its hydrogen, which makes it an acid or a
So
proton.
if we were to write it out, let's see.
It'd be HHN, carbon, and then the acid group is up here on the far side.
Or does it have?
To draw it out.
Because I don't memorize the peptide bonds. Okay. An amino acid looks like this.
You have 2 hydrogens, and your hydrogen what?
It's carbon, right?
Yeah, it's carbon in the middle with the hydrogen and an R group.
Carboxylic. And an R group. R group means and something else.
Right, like thymine or
It's
or
usually what makes
glycine,
them.
whatever the amino acid
Usually
goes
it's
in
a
there.
chain, but yeah, there's something else attached. That's an amino acid.
The amino group is from carbon, oxygen, and hydroxy. And the
So
other-
this is an amino group. This is a carboxylic acid group.
Got
Okay. Okay. What do proteins do?
They're the building blocks of the body.
Okay, building blocks of body. Okay. What are some functions that you know with some proteins?
Some
gives energy.
Energy. Yeah, proteins can
become enzymes.
proteins can be broken down for energy. They're enzymes, so that's really, really important. So catalysts, they're able to lower the activation energy. What else?
Like the body
Transport,
process.
protection.
Transporters, so protein transporters. There's a lot of things. More or less, protein's really important. They're able to help with protection. You can break them down for energy, transporters. I was about to say
Structure.
something else. Structure,
Construction.
and enzymes.
Regulation.
Oh yeah. Yeah, really important. They're sensitive to pH, which is another topic that you guys want to talk about. So very sensitive to pH, so you can't have the pH vary too much from what it's set at within our body, or else you're gonna mess up with these proteins. Okay, so proteins, again. So there's the folding of the proteins, okay? So there's 4 different steps, if you will. So if you can remember any class where you learned about DNA and whatnot, RNA comes out, it's gonna be translated, right? So the ribosome latches on to the RNA strand and starts to translate a polypeptide or an amino acid chain. Okay? So it's putting these amino acids together, forming a protein. Okay?
Okay.
So first, it's going to be pumping them out in just kind of a chain. Yeah, I'm just gonna save a lot of time.
Look at that incredible protein chain.
Yeah, that's a great protein chain. Okay, and so the only bonding that's going on right now is going to be between.
And so that makes it the primary structure because it's just the.
So the primary structure is before any folding or shaping of the protein has really taken place.
Okay.
The only bonds that are in action right now are going to be covalent bonds between the different amino acids. Okay.
That helps.
Okay, so that's the primary structure. Then, because we're dealing with molecules and they get affected by everything, you're gonna get your secondary structure,
Which is the spins and the flat.
which either looks like a helix, which that's the best I can do with a helix. Double helixes are so much easier to draw. Or pleated, like folded sheets. That should be in your book, that's on page 47, I think. Yeah, so it's pleated. And why does it say that happens though? Hydrogen bonding.
Yeah, okay, I
Okay.
see.
Okay,
Yeah,
you can
okay.
even see that with
Yeah.
the red dotted lines between the hydrogens and
And that's
oxygens.
what causes it to bleed.
And so that causes it to pleat. So it starts to get a little bit of shape because so the chain itself starts to have so here's a hydrogen right here, oxygen, lots of hydrogen bonding, okay? And so it starts to fold. You're gonna get your tertiary, so this was secondary structure, right? So you're gonna get your tertiary structure. Why? What causes that?
It's more of a large scale. It's this at a larger scale, almost like this continues to print out. This is what's great because of it.
So what this, I know it just looks like a beautiful scribble because that's kind of what it starts to look like. It starts to fold a lot more directions because of interactions with its environment. For example, this is going to be, we'll say this is one of the ones that's happening out in the cytoplasm. Sometimes it'll happen inside the Golgi and you'll see that. But in the cytoplasm, there's a lot of molecules floating around and they have a lot of interactions, and so it causes it. So say that this is a non-polar portion of this protein right here, and here's a polar molecule coming by, and so it pushes it away, but this guy's attracted to it, so it yanks it over here, and so it starts to make a whole different shape,
Yeah.
okay?
Okay.
And then you're gonna get your coordinary structure when you add those subunits together, okay? So by subunit, meaning here's a subunit, here's another subunit, here's another subunit, and here's another subunit. So these 4 subunits, which was alpha 1 and 2 and beta 1 and 2, come together and made hemoglobin, was an example that you talked about. So hemoglobin is made up of those 4 subunits, which they interact with each other and cause more folding.
Okay.
And so you got your primary secondary, tertiary, and quaternary structures. Primary structure is just gonna be the covalent bonds between amino acids.
Yeah, with the printing of the,
Secondary is Y,
the hydrogen bonding.
hydrogen bonding. Tertiary, Y,
It's the tertiary is the environment around it.
the environment. And quaternary,
Quaternary is between other tertiary.
yeah,
groups
so
or
other
much bigger.
tertiary subunits, tertiary folded subunits all come together and interact with each other, causing more folding, which result you with a protein.